So my name is Sean Megason. I'm an assistant professor across the river in the Department of Systems Biology at Harvard Medical School. And going with the central dogma organization of this meeting, we've kind of worked our way up from DNA to RNA to protein to cells. And finally, this session is about visualizing organismal data. And we're going to have 3 talks, which will hopefully give you some insight into kind of the different stabs at attempting this. I would say from the start that I think just the nature of the problem is quite difficult. And yes, we're visualizing organismal data, but we can't leave behind all those other scales that we've talked about along the way in this conference. So information on genes is still important, on RNA, on protein, on cells. All of that kind of has to come together to make an organism. And importantly, and what I've tried to convey in the title of this talk, is that we have to understand that not just at an endpoint, but at least for my interest, which is understanding development, we have to understand that across time as well, developmental time. So in the case of visualization, it presents some significant challenges just because of the dimensionality of the data. So I come at this question from a basic science point of view. I'm a biologist, and at the end of the day, my goal is to discover new biology. And the basic question I'm interested in is how is life made? And for me, the key step of that is understanding how we go from a fertilized egg, shown here, to a fully patterned embryo, which will then continue to grow to make the adult. And we ask this question, but I think it's important to ask what we actually mean by this question. So what constitutes understanding? And this is in many ways a difficult question to ask, but we can start to pick at it and get some ideas as to what meaning might mean. So kind of a classic example in development is the eye. And it's known that there's this gene PAX6, which is necessary for making eyes. And it's also needed in Drosophila for making eyes. The gene's called iris. And it's also sufficient. You can misexpress this gene in many cases and get eyes in other parts of the body. And for many people, that kind of constitutes understanding. We have this gene. It's necessary and sufficient for making an eye. Therefore, we understand how eyes are made. I would argue that that doesn't really constitute understanding. Because it's too reductionist. It's kind of silly to think that this one gene makes an entire anatomical structure. And it's too molecular. So when we ask what mechanism is, a lot of people argue that molecules are mechanism and mechanism is molecules. But what I want to argue is that mechanism happens at many different scales. And to gain mechanistic insight, we need to look at many scales. We need to be able to visualize data from many scales. So what do I mean by it being too reductionist? I would argue that the mechanism involves a system. There's not some linear pathway that goes from one gene to one phenotype. In reality, there's some type of network that's underlying that phenotype. And oftentimes, what's more interesting are the motifs and the connections in this network and the feedback rather than exactly what the nodes are. I would also argue that this kind of thinking is too molecular and that mechanism happens at many scales. So molecules are certainly important, and how molecules form networks is certainly important. But there's a number of other mechanisms that happen at higher scales. So at the cellular level, in development, cells can adopt different fates. They can divide, they can die. These are all cell-level phenomenon, which are best abstracted at the level of the cell rather than above or below. There's also what I call the supracellular level. So there's a number of mechanisms such as lateral inhibition, cell intercalation, group-wise migration that occur at the supracellular level that are best understood by abstracting and visualizing data at this scale. Of course, you can work your way up to tissues. Tissues will bend and grow and finally form organs. And of course, all this is happening in order to make an organism. And there's different questions from a biological point of view that can best be thought of at these different levels. So again, mitosis, apoptosis, and cell specification, yeah, you can probably understand them pretty well at these lower levels. And I would argue that these are actually what we better understand in biology. There's other questions which people have been interested in for a long time, but unfortunately are not as well understood. So for example, patterning, how do you get the right types of cells in the right place? Morphogenesis, how do you form the right shapes of tissues? And size control, how do you get tissues and organs to be the right size? And I think these are not as well understood because the answer, the mechanism actually spans all these different scales. So it makes them just fundamentally a more difficult question. And likewise, there's different approaches for analyzing these. Genetics and genomics is great for the molecular kind of network scale, but not as good for these higher scales. Modeling, which I think has a lot of important connections to visualization, is also important, but typically done in different fashions for these different scales. And what my lab works a lot on is imaging. And the reason we focus so much on imaging is because imaging can actually address all these different scales. So we can fluorescently tag molecules and image single molecules. We can color different nodes of a circuit in different colors and see how these colors go up and down as some process is being computed in development. And we can track cells, we can track tissues, and we can do all this in the context of a living, developing organism. So these are all across space, but the important thing about development, and in my mind the important thing about understanding for many biological processes is time. So for me, understanding means what makes things change over time. And imaging can give us this time, but then visualizing data over time, it's one more dimension that makes things more difficult. So we can think of this as two different dimensions. So we have scale from atoms to organisms, and we have components from one to many or all. So developmental biology, we've been doing visualization for a long time. We'll do antibody stings or in situs. It's typically one gene, and we're able to visualize that. I think more recently, in the past decade or so, with systems biology and the various genomic and proteomic approaches, we're now capable of measuring all the components, and a lot of the talks at this conference have been about how we can properly visualize many things, because visualizing many is different from visualizing one or a few. I think the problem that I've been struggling with and my lab's made some attempt to deal with is this kind of much bigger chunk of the graph. And these questions I talk about of patterning, morphogenesis, and size control fall within this realm. So the scale is from genes to the whole organism, and there's many components that are involved in accomplishing these goals. So how do we visualize something across many scales and across a large number of components? So as I said, the principal approach my lab has taken is something that we termed in toto imaging. And the idea of in toto imaging is to try to image things completely in toto. And the way we do that is using zebrafish, which is shown here. And we've developed a number of transgenics, which will mark things in different colors. And then we can just, if the goal is to understand how you get from egg to embryo, we can simply watch. So that's what's going on here. So we start at the one cell stage. Mostly what you're seeing now is the cells are dividing. We've labeled the nuclei in red and the cell membranes in green. This is cleavage stage. The cells are about to undergo a process of gastrulation in which there's a lot of cell rearrangements. So we can, with our imaging, we can image and capture an incredible amount of raw data. And there's very serious problems in how we understand this data. So these type of in toto image data sets are typically 100,000 images, 3 megabytes apiece. So data-wise, there's a huge amount of data. And what I hope you'll take away from this is that there's an incredible amount of cell movement that's going on. What's not shown here is that, of course, genes are going on and off in all these different cells. And presumably, the gene activity is what's controlling all these different cell behaviors. So the real challenge is linking how genes are turned on and off with controlling cell behavior. And I would also argue that understanding how other forces besides molecular forces, such as physical mechanical forces, are organizing this process as well. So I guess an obvious practical aspect of data visualization is just what software tools. And I decided not to be too preachy about this because I think it's an unsolved problem and there's lots of tools. And I'll just say which tools we use and mention a few others. We've put a lot of effort into developing a software package called GoFigure, which is basically to support in toto imaging. I'll talk more about that. We've developed a number of algorithms, some of them recently published, one called ACME for doing membrane-based segmentation. And then for just straight up visualization, we use a nice program called Flow Renderer from University of Utah. There's a number of other packages which are quite relevant for this type of data. ImageJ probably has the longest history and is the most well-known. Fiji is an implementation of ImageJ or packaging. There's a fairly new package called ICE from the Pasteur Institute based on Java for doing image analysis and visualization of organismal scale data. And you've probably heard some about Slicer, and I think the following speakers will talk more along those lines. Slicer was mostly developed with the ideas of medical imaging in mind, but the problem certainly relates to organismal scale data in general. So I'll talk more about GoFigure, since that's what I know the most about. The goal of GoFigure was to simplify in toto image analysis. So there's a couple of tasks that are important for doing that. So one is just manual segmentation and tracking, because automated segmentation is never perfect. But we've also put a lot of effort into doing automated segmentation and tracking, so recognizing cells and tracking cells in these movies I just showed you. Information visualization is certainly important. There's a huge number of dimensions, and it's not trivial to find patterns in the data, but by color coding the data different ways, that can help for analysis. And this software is based on a lot of common packages, which you guys are probably familiar with. So Qt for the graphical front end, the visualization toolkit for doing a lot of the visualization, the Insight segmentation registration toolkit, and then all the data is actually stored in a database using MySQL. So I've taken just a couple of movies of GoFigure in action to try to show you just some examples of how we approach this problem of visualizing this data. So as I said, the movies are at least 4 dimensions, so X, Y, Z, and then time. So simply being able to navigate through these very large image sets of 100,000 images is quite useful. And that's what's shown here. There's kind of a fairly standard view of having what we call these orthogonal sections, so X, Y, X, Z, Y, Z, and then the 3D view. And you can easily switch in between those. And of course, you can visualize the different color channels as well. And something I'm not sure if it's shown here, but there's also what we call a Doppler view in which we color code time. So we can superimpose different time points, and that gives you a feeling for how cells are moving over time. The data model for GoFigure is hierarchical. So we've divided it into what we call contours, which is what is being drawn here. And a contour is a 2-dimensional representation of some biological object. It could be a nucleus or a cell membrane or an organ. And there's tools for doing fully manual segmentation, or what we call semi-automated segmentation, which was just shown here, in which a seed point was put in a cell. And then we have algorithms for automatically segmenting out the nucleus that seed point was in. And then since automated segmentation is never perfect, it's important both to be able to visualize the results of automated segmentation, so you can, by eye, go through it and see what's correct and what's not correct, and then also to be able to correct it. So there's tools for correcting segmentation of these meshes, which is a 3D object, as well as tracks, which are 4-dimensional objects, so connections of meshes over time. So here we're correcting tracks. And then the final step is what we call lineages. So lineage is a collection of tracks over cell division, from mother track to daughter tracks. So what we're showing here are meshes. So these are cell segmentations that were performed automatically on a cluster, because all these algorithms are fairly slow. And then we can import that data into GoFigure for visualization. So you can see in the 3D view here, we're just rotating it to get a better sense for how these cells relate to each other. In this case, we've just randomly color-coded the cells to help distinguish them. And then you can go through and pick cells using a 3D picker. And then this table view at the top will show you various parameters about that cell. So it could be the size of the cell, the position of the cell, the velocity. We can also image transgenic zebrafish, and then these columns would represent the levels of gene expression. So now we're starting to span these different scales from gene expression up to cells and tissue. Okay, I'll stop it there. So the next level of the hierarchy are these tracks. These tracks were also done in an automated fashion, and the tracks represent the movement of cells over time. Once you have these tracks, you can start to visualize how different cells move differently to form tissues, and you can color code them in various ways to try to help your eyes make sense of this big spaghetti bowl. So we can color by time, so just going from red to blue shows the progress of time to help you give you a feel for how cells are moving, or you can color code by speed or angle or by anything else. I guess I skipped over it on the last slide, but likewise on meshes, you can color code by gene expression, by size, by anything else. And that can help your eye find pattern in this complex data. And then finally, lineages. So lineages, a lot of you guys have probably seen the icon of the C. elegans lineage tree. So that's a lineage tree that shows topology, but it doesn't show geometry. So in GoFigure, you can view lineage trees geometrically embedded, so meaning the tracks actually are shown as they are in the real-world coordinates of the geometry, or you can remove the geometry and just show the branching pattern between mothers and daughters. So you can edit these tracks, and then you can color code by depth or breadth or various branching patterns as well to help your eye see if there's different lineage motifs that might give rise to different types of cells. Okay, so that's kind of the front end. And we mostly think of GoFigure as a tool for interactive data exploration. And then segmentation can be done offline. So we've developed various tools. This one's called ACME, Automated Cell Membrane Extractor, which lets us do segmentation of these cells. The way ACME works is shown here. We start with a raw image in which we've made a transgenic zebrafish that has a fluorescent protein that's localized to cell membranes. The basic idea here is that the concept is that membranes are locally planar. So by taking the Hessian, the second derivative of the image intensity, a plane will be consistent in two directions and rapidly changing in one. So we can take the Hessian and then look for that pattern to find planes and then enhance planes. So we get what's called a planarity image. And then we can do something called tensor voting, which is basically to fill in gaps that might be caused by image noise. So we get a reconstructed image. And then once we have a reconstructed image, we can do fairly basic segmentation algorithms such as watershed to now get full segmentations. So now we're able to do segmentations using only membranes, only nuclei, or in really difficult images, we can combine the two. And that frees up some channels for tracking gene expression or other types of data as well. So once we've done that type of segmentation, we can now visualize the data in a different way. So you're probably normally used to looking at volume renderings. This is not a volume rendering, it's a surface rendering. So we've automatically segmented out all the surfaces, And now we can render them as surfaces rather than rendering the raw pixel data as a volume. And this is nice in that it allows you to better adjust the opacity so you can see through cells. And since, of course, the data is all digital, you can go through and do 3D picking to pick which cells might be interesting for you. And then back in GoFigure, it would highlight those cells, and you could see what's special about those cells. Or you can do these kind of virtual dissections or virtual cell ablations that I just showed. Also within GoFigure, you can look at segmentations of nuclei. So these nuclei were done automatically. I would say all this work, there's some biological reason we're doing it. Here, the biological question is how do the surface cells become a different type of cell than the interior cells? So the surface cells here are shown in gold, and the interior cells are shown here in blue. And the question we're asking is, when do these cells become different? To ultimately understand why they become different. And the basic idea, the basic hypothesis we're working with is that it's actually the shape of the cells is the first thing to change, just for mechanical reasons, because they're on the surface, they're getting pulled by surface tension, and then that change in shape actually induces a change in fate. And by doing these kind of visualizations, we can start to explore those type of hypotheses. We've done a lot of work on tracking and lineaging as well. I'm kind of embarrassed standing in front of Felice. It's both intimidating talking after Felice, but also useful. It's intimidating because I feel that everyone's criticizing my slides as I go. But it's also useful because I want to talk a lot about some concepts that she very beautifully introduced. So this is a lineage, which by the nature is kind of a spaghetti bowl of data. So these lines are showing the movements of cells over time. They're color-coded, so we start at red at the 2-cell stage, and you can kind of see how each of these cells divide orthogonally and move out into 4. Those 4 divide into 8 and move out in an orthogonal direction. And here we're actually inside the embryo looking up into the embryo. And now we'll rotate around, and hopefully eventually your brain will be kind of able to sort through the basic shape and pattern of what all these cell movements are doing. So here, what we want to understand is how cell division, orientation, and size of cells is controlled to ultimately build the embryo. And it's a very complex process, so being able to visualize many time points at once using something like this is useful, and being able to rotate things around to try to get a feel for how cells are moving is useful. Okay, so that's kind of it for intro to GoFigure. I wanted to transition to kind of how do we actually use this to discover new biology, and also kind of talk more about some of the points that Felice brought up, which, I mean, I guess is the following. So, you know, visualization is certainly very important for communicating data. I think it's also very important for understanding data yourself. So I think primarily visually, I think somewhat mathematically and somewhat textually as well. But when I'm trying to figure out what my data means, it's actually useful to be able to visualize it as these renderings, but also to be able to visualize it in models. So it's certainly useful. I think everyone here would agree with that. But visualization can also hinder our thinking as well. And that's this idea of iconography and paradigms. So we have a paper that's coming out in a few weeks in Cell, which is addressing this long-standing model. The icon is shown here, and if you open up any molecular cell biology textbook, you'll see a picture that looks like this. And the point of this picture is looking at how pattern is formed in the neural tube. And the textbook model is that it's patterned by a morphogen. So there's a gene that's expressed here. It makes a protein that diffuses. Cells then look at the concentration of that protein, and they adopt different cell fates as a function of concentration. And since concentration maps to position, you get different cell fates in different positions. So that's the textbook model for morphogen patterning. There's a lot that's right about this model, but there's some things that are wrong about it too. And it's difficult for people to see what's wrong about it because this icon is so satisfying and so intuitively easy to understand. What's difficult is that in development, you don't just have a blank slate of a neural tube that looks like this and then you pattern it. you actually have to build this structure over time. So this structure is constantly changing over time. It's growing, it's undergoing morphogenesis, and this patterning is happening at the same time as these other processes. So visually, it's just very hard to show what the neural tube looks like as a function of space and time because it's constantly changing. So kind of the point of this paper, which I won't really go into the biology, but I'll show you our attempts at kind of how to understand this visually. So basically what we do is we image the neural plate of a zebrafish over time as it forms, and we're able to watch every single cell in the neural tube as the neural tube is formed. And then we process the images and track the cells. So the movie looks something like this. It's a four-dimensional movie. Here we're looking at it from a top view, looking dorsally down to the neural plate. And then here it's the same image set, but we've just taken a cross-section of it to start to watch how the neural plate forms the neural tube. So what you can see is that there's a lot of cell divisions, there's a lot of cell movements. The cells are basically piling into the middle here to form the neural tube. This is a notochord shown here, which is making this morphogen, and the somites are on either side. And eventually what you see is this third color, green, come up, and this is marking the fate of one particular type of cell, which are motor neuron precursors. But it's just to give us an idea of what the fate of one of these cells is. We can then track all these cells and track them backwards in time to see where they came from. The point of all this is that they don't really follow the morphogen model, but it's kind of hard to show you exactly why not. So we've attempted this in a number of ways. One is just these diagrams showing you that the neural tube has to be built over time. So initially it's a plate, those cells squish together to form this neural kill or neural rod, and eventually it forms a neural tube. That's one way of showing it. I won't go through this, but we've done a lot of other types of presentations to show how cells move over time, how sister cells related to each other, just to give people a feel for the movement. Perhaps a more useful way of showing the data is just, again, this geometrical view, combining the images, which we have different orthogonal planes showing the neural plate and the notochord, and then these spheres are cells that we've segmented and been able to identify what type of cell they are, and then we've color-coded the cells by type. So the goal of the neural tube is to make a nice pattern where you have different types of cells in the right position, and you can see there's this very nice, precise pattern of different cell types. But if we rewind the clock back in development, we can see that this pattern is all scrambled together. And we can also visualize it in the following way, in which we have time and distance from the notochord. And eventually you get a nice pattern. So again, here we're color coding cells by type. From red to blue, they form this nice organization. But if we look earlier, the cells are all scrambled up. And we've done a lot of tests that I can't show you that show that these cells are actually specified at this point in time. And that the way that you generate pattern in the neural tube is not just by a morphogen. The morphogen will give you this kind of pattern, but it's then by the cells sorting and moving into the correct pattern. And then you can also visualize this in a movie, which of course you can't show in a journal article, but it's useful for presentations such as this. Again, we're tracking two cells color-coded by type. The point of this movie is that those cells start in the opposite position that they should be, and then by the end of the movie, the cells flip around and rearrange with each other, such that the floor plate cell is closer to the notochord and the motor neuron cells further away from the notochord. So these are all different ways of visualizing the raw data. You sort through enough of this raw data, you try to form a model in your head, and then you try to represent this model such that you can communicate it to other people. Basically, the old model is shown here. There's a long-standing tradition of the iconography in the field of using a French flag. It's called the French flag model. The old models, you have a nice, smooth morphogen. It then generates very precise stripes of expression. The new model, we're kind of also talking in analogy and metaphor to this earlier model, but we're emphasizing that just the positional information, the morphogen signaling is noisy, and therefore cells are specified in noisy position, and then there's this mechanism of self-sorting. It's a cellular mechanism, it's not a molecular mechanism, that allows you to go from a messy pattern to a nice pattern. And I guess a lot of you guys have seen that there's a lot of journals are now having these visual abstracts, which is kind of a different way of communicating information. So this is our attempt at trying to explain this model with new iconography. So here we're showing cells, which are the circles. They're moving in time. Again, how do you show time in 2D? There's a morphogen, which is the triangles forming a gradient. So here we're trying to emphasize the dynamics. Here we're showing the cells getting specified in a not completely perfect pattern, and then the cells move to form a nice tight pattern. So that's the way we try to go from a lot of data that doesn't make sense even to us to try to form models for ourselves, and then finally to try to present them to the wider scientific audience. And I wanted to go through this exercise to show you that, or to hopefully argue that, Felice showed this example of, the chimpanzee turning into the human. We can laugh at that, it's kind of obviously wrong, but we also delude ourselves in our science. The nature of science is you get new data, it doesn't make sense, you try to fit it in with old models. It's sometimes hard to distinguish how much of those models is truth and how much of it is imagined. So we need to constantly question our own models to figure out how our new data fits into it. So I would just caution to be careful about any type of model or any type of icon in science, and maybe it follows the chimpanzee example as well. So the people that actually did the work, there's a computational group shown here that developed the software of GoFigure and these various algorithms we use. My lab basically studies the neural tube and the inner ear, which I didn't talk about, and then the final work I showed you about pattern formation through cell sorting was done as a collaboration with Alex Shear here at Harvard and a postdoc of his, Peng. I'll stop there. Thanks a lot.